Well, good morning. Thank you for being here. My presentation will deal mainly on post-harvest aspects of fruit cuticles. I'm very glad and grateful to have the opportunity to show you a part of our role, of our work on this topic. And yes. So the cuticle itself is not a novel topic. Its existence has been known for a long time, actually for centuries. For example, in this botany treatise from the 18th century, it is described as a structure that does not dissolve when plant tissues are macerated. And this description is very interesting because it clearly reflects the mainly hydrophobical nature of its main chemical components. The main chemical components of fruit cuticle, of plant cuticle, are usually classified into two major types. So the cuticular waxes, and then the catin, which is a polyester. To make it simple, we can say that it's the matrix where the waxes are arranged. And sorry, and cuticle composition, the chemical cuticle composition, is highly variable among different fruit types. For example, I will show you some of our results in a range of fruit. When we began working on this topic, we did a survey of published literature on cuticle composition, specifically in fruit species. And well, actually, we found the main idea we obtained after this review was that the available information was actually very limited. And in the cases where it was present, it was only partial. It was not complete. Of course, we found information for tomato, which is the typical model fruit species. And then we found some information for apple, for pepper, for sweet cherry, some citrus fruit types, and grapes, but actually not much beyond that. So first of all, we had to characterize the chemical composition of cuticles of the fruit types we were working on. And in this slide, I show you the main wax compound families or wax compound types that we identified in different fruit types. For peach, we considered a melting and a non-melting peach cultivars, and in both cases we found three main wax compound families, namely the triterpenes, then the alkanes, and then the alkanoic acids, the monocarboxylic acids, with minor amounts of other compounds. Well, even though the main wax families were the same, please note that there were important differences in the relative amount of these wax families between both cultivars. Sorry. Oh, I'm going backwards. Okay. For sweet cherry, we also considered two different cultivars, and well, the main wax families were the same and more or less in the same proportion. The main difference was that, as you can see, the total wax coverage was much lower in sweet cherry than in peach. It was around 10%, that in peach. For blueberry, we have data only on the triterpenic fraction. So, well, the total amount of the relative amount of triterpenes in blueberry was around 45%, which is quite in accordance with data on peach and sweet cherry. But the situation was quite different for kiwi fruit. In kiwi fruit, we studied green flesh and yellow flesh cultivars, and we found remarkable differences in comparison with the previous cases I have shown. To begin with, you will see that the relative amount of triterpenes was extremely low. Then there was a huge amount, I would say, of alkanoic acids, of monocarboxylic acids, and there were also important amounts of phenolics, which we had not found in any other fruit type. And we also analyzed the cuticles of olive fruit. Here I show data for two cultivars. This one is Arbequina, which is the main olive cultivar in Catalonia, and the other one is Manzanilla. And we found some interesting results because, for instance, as you can see, there was an enormous relative amount of triterpenoids, but the main difference was the very low amount of alkanes. And I say that this was quite a surprise. It was unexpected because, as you may know, alkanes are often related to the waterproofing properties of cuticles. And olive tree together with grapevine are the typical historical crops in the Mediterranean area. And incidentally, in the Mediterranean area, the annual rainfall is not very high. Of course, it may show some differences according to the specific site, but in general terms, the rainfall is rather low. So it was quite a surprise to find such low amounts of alkanes. When we analyzed the cutting of the same fruit types, in the case of peaches, we found three main monomer families, so the mono and the dicarboxylic acids, and also a high amount of hydroxy acids. Similar results were found for sweet cherry, so the three main compound types were the same and in similar proportions and similar quantities. After all, both sweet cherry and peach belong to the same genus. They are Prunus species, so they are very related. But the situation was very different for blueberry. You see that, we again had around 1/3 of monocarboxylic acids, but instead of finding dicarboxylic acid, we found another third of alcohols, and the relative amount of hydroxy acids was comparatively lower. And in the case of kiwi fruit, we found important cultivar-to-cultivar differences. As you will see here we found a role very predominant for hydroxy acids, but please note the huge or the remarkable differences in the relative amount of hydroxy acids between both cultivars. And then for kiwi fruit, we also found a big amount, particularly for sorellis or the yellow-fleshed cultivar, a large amount of compounds that we could not identify in that moment. So we don't know what this is, but it is quantitative important. And finally, for olive, we found very high amounts of total cutting in these fruits in comparison with the other cases. Please note that this axis is very different to this one, because in general, cutting loads in olive were at least 3 times higher than in the other fruits we have studied. And and as you can see, we also found very high relative amounts of hydroxy acids. So what I have just shown corresponds to fruit at harvest. But cuticle composition is not static. It changes, it evolves. It evolves during maturation and also after harvest. So let's have a look at some examples of these changes in different situations. In the first place, we found that fruit cuticle composition is modified after harvest, both if you keep fruit at room temperature or in cold storage. For example, in this experiment, we stored sweet cherry fruits at 0 degrees for 2 or 4 weeks, and then kept them at room temperature for 3 more days in order to simulate commercial shelf life. And we found important changes. We found important changes in total cuticle load, both immediately after removal from cold storage and after the shelf life period. These changes were not only quantitative, but we also found some changes in some values and some indicators, such as, for instance, the wax to cutting ratio, the average chain length of the acyclic wax compounds, and also in the ratio between 16 carbon to 18 carbon cutting monomers. When we expressed this data not as a percentage, but in terms of quantity per surface area, we found a significant increase in total wax amount 3 days after harvest in non-stored fruit. So 3 days after harvest. But however, wax amounts were substantially lower during and after the cold storage, with no large differences in the relative amounts of each compound type. Concerning cutting, we also found this increase 3 days after harvest in non-stored fruit. And regarding cold storage, well, there was not a clear trend, and the changes appear to be more related to storage period. So, in the second place, we have also found that fruit cuticle composition changes in response to some post-harvest treatments. In this experiment, we submitted peach fruit, specifically melting type peach fruits, to heat or to carbon dioxide shocks, and then we kept the fruit under cold storage for 2 weeks, and then during 5 days at 20 degrees. And again, we found changes in total cuticle load, both immediately after removal from cold storage, but also after the shelf life period, particularly for heat-treated fruit. When we analyzed the waxes of this fruit, we did not find any quantitative or qualitative differences upon removal from cold storage, regardless of treatment. However, we did find these differences after the shelf life period, particularly for heat-treated fruit. And as you can see, the relative amount of triterpenes was high anyway, but decreased, which led to higher ratios of acyclic to cyclic ratios in this fruit. Regarding cutting, the trend was the opposite, because as you can see on this slide, there was an increase in total cutting load immediately after removal from storage, but not thereafter, with the exception, again, of the heat-treated fruit. So it appears that cuticle of this fruit reacted differentially according to the specific treatment they had received. And well, the most important question to ask is, how do these differences or changes impact on fruit quality? That's the most important thing. We did a review of published literature on fruit cuticles from the post-harvest point of view. And based on this information, we identified 4 main aspects of fruit fruit quality susceptible to be impacted by cuticle properties. The first one, of course, is water loss. Cuticles restrict transpirational water loss, and this is actually the main role that has been historically attributed to plant cuticles. Actually, the plant cuticle evolved as an adaptation of plants to the terrestrial environment. But fruit cuticles are also related to the susceptibility of fruit to biological attack and to some surface disorders, such as cracking or russeting, for instance. And also, the cuticle confers the fruit with particular mechanical properties, which means that the cuticle might be helping modulate textural properties of the fruit, such as firmness, for instance. So let's have a look at an example of how cuticle properties may be impacting water permeance of cuticles. We did a study on 9 different olive cultivars, and in this slide I will show you data for 2 of them, of these cultivars, uniquely, namely Farga and Sevillenca. To begin with, please note that the total cuticle load was the same in both cases. But however, there were important differences in cuticle characteristics. For example, wax to cutting ratio was much higher in Sevillenca than in Farga, and the thickness of Sevillenca cuticles were also higher. So Sevillenca had thicker cuticles. As regards waxes, Sevillenca had also more waxes, both in quantitative but also in qualitative terms, so in percentage. And Sevillenca had also more cutting. In this case, the differences were not so large, but they were significant anyway. In addition to analyzing the chemistry of fruit cuticle, we also evaluated some surface properties. And you can see that we found important differences in the fruit surface topography. In the case of Farga fruit, the surface of this fruit displayed a high degree of roughness, while the surface of Sevillenca fruit was smooth, and besides, this fruit stained in the toluidine blue test, meaning that they displayed some kind of discontinuities or porosities or little cracks or whatever on their surfaces. However, and in spite of this difference at the chemical and at the surface level, the water permeance was the same in both cases. Well, it could be that these differences were not large enough in order to reflect on water permeance. So please let me show you another work, also another experiment, also in olive, which we did in collaboration with Universitat Würzburg in Germany. In this case, we analyzed both fruit and leaves of Arbequina olive. For fruit, we considered 3 different maturity stages. So we had green fruit, turning fruit, and black fruit. And well, you will see that there was an increase in total cuticle load during maturation of fruit. Okay, it was not a sharp increase, it was not a dramatic increase, but it was a steady, sustained increase. There was also an increase in total wax and also in total cutting. And well, it was not only a matter of quantity, because we also found differences in other values, such as the acyclic to cyclic ratio and the average chain length of the aliphatic wax compounds was longer, and the cutting monomers were also longer, as indicated by this ratio. However, when we determined the water permeance, there were no significant differences among all 3 maturity stages. So next, we analyzed the leaves. We analyzed in specifically the adaxial side of the leaves, and you will see that there were important differences. First, the wax to cutting ratio was much higher in leaf than in fruit, regardless of maturity stage. It was more than double the values in fruit. total was there was much more total wax, was both in absolute and in relative terms, and there was much less cutting. As you will see here, the total cutting in leaves was about half that in mature fruit. When we determined the water permeance of leaf cuticles, we found that these cuticles from leaves were less permeable to water than fruit cuticles, in spite of something I would like to highlight, in spite of the fact that total cuticle load in leaves was much lower than in fruit. So I would like to use this data to make a point here. And my point is that some people usually assume or give for granted the what I call the more is more model. The more is more model. So these people assume that the more cuticle, the more thickness, the more waxes, the more alkanes, the more anything will result in better waterproofing properties. But according to experimental evidence, I think that this model is probably a little bit simplistic. So it could be true in some cases, I do not deny that, but the experimental evidence indicates that this is not in many cases. Of course, the compounds have to be there. So some quantity has to be there, that's clear. But there must be other factors that help modulate the water permeance of cuticles. For example, wax to cutting ratio might be one of them. Regarding waxes, the acyclic compounds are often considered to favor the assembly of waterproofing barriers. So the higher amount of acyclic compounds and the longer the chain of these compounds should be favoring the waterproofing function. Concerning cutting, hydroxy acids and longer cutting monomers are also considered generally to favor a correct assembly of the cutting matrix. So this should favor the waterproofing properties of the cuticle. And there could also be other factors involved. Structural factors, morphological factors. For example, these images shows the cuticles of 8 different tomato genotypes, and you can see that there were important differences, morphological differences between them, among them. So regarding thickness or roughness or the degree of embedding or the presence of particular morphologies. So this could be one more factor which could help modulate water permeance of cuticles. So what can conclude for all of this? Well, the first thing we can conclude is that we do not know many things. So the first conclusion is that there are many things that we do not know. And this involves, I would say, 3 levels. The first level of things we do not know is related to the simple description of facts. So for many fruit genotypes, the cuticle composition has not been described, or at least has not been reported. So this information for many fruit genotypes is simply non-existent. Then we would also need to know how this composition changes during maturation, after harvest, and in response to external factors. Then we would need to know how these changes and these differences are related. Then, which is the relationship between composition, so the specific compounds, the structure, and the function, and eventually how this results and how this impacts on fruit quality attributes. Regarding the impact of cuticle properties on quality attributes, there have been also some reports. Most of these reports are merely quantitative. So, for example, it has been reported that higher loads of total cuticle had this or that effect in a little choice of fruit types. Sometimes we will find some surprises. For example, it was reported that higher cuticle loads resulted in higher water loss in pepper, which according to the Morris-Moore model should not be expected. There have been also some reports on some cuticular wax types, on different aspects of fruit quality. And well, I would like to use this part of the table to show you that sometimes we could and actually we will find contradictory results, opposite results for different fruit types. For example, it was reported that higher amount of esters led to higher water loss in blueberry, but not in grape. This means that we will probably need to consider or to study each fruit type individually on a fruit-by-fruit cases. So? And well, there has been also some reports on total amount of cutting, which generally relate to resistance to biological attack. And in relation to qualitative traits, there have been also some observations that this ratio between alkanes and triterpenoids and esterols, well, it's actually the ratio between alkanes and cyclic compounds. This ratio has sometimes been reported to be related to lower water loss in a few examples of fruit, sweet cherry and peach. We found that relationship ourselves in our works, and there was also a report on pepper. So, well, we have many things that we do not know. We have some reports, partial reports, but we also have a few facts. So many unknowns, some observations, and a few facts. The first fact is that there are important differences in cuticular properties among species, cultivars, maturity stages, and also harvest. The second fact is that fruit cuticle composition is modified after, well, during cold storage, and we should keep in mind that the cuticle is highly reactive to the environmental conditions. So it is highly reactive to the surrounding conditions. And in the context of cold storage, the main factors are, on the one side, relative humidity and then temperature. Any of both factors will cause changes in the biophysical properties of the cuticle, such as rigidity or elasticity or something like that. And these changes in the biophysical properties of the cuticle are highly likely to affect the mechanical resistance of the fruit. So again, it could be that through the modification in these biophysical properties of the cuticles, we have a modification in the textural properties of fruit. And finally, another fact is that fruit cuticle composition is modified differentially in response to particular post-harvest treatments. Of course, we have not assayed all possible post-harvest treatments, but the cuticle is modified to all in response to all of the harvest treatments, post-harvest treatments we have applied. So this is all from my side. Thank you for your attention. I hope you found it interesting. Thank you very much. So
I have a question. Do you think that there could potentially be a path to modifying cold storage treatments in order to have a specific reaction to the cuticle for an improvement of food quality, or whether related to pathological infection or something like that?
Well, as far as I know, my research experience indicates that it's very, very easy to modify the total amount of cuticle, the total amount of waxes. But the other thing is if this, what happens with the composition of these waxes. Maybe you have more wax load, but maybe you don't have enough triterpenes to sustain. Oh, yes. Sorry. Yes, I was saying that it's quite easy to modify the quantity of cuticle load, but a secondary point to look at is if the composition of this cuticle would be the right one for the property we wish to modulate. So the biochemical, the metabolical routes that go to the triterpene synthesis are different than those for the aliphatic compound synthesis. So it could be that you have more cuticle, but it's not balanced, to say something.
If I understood right, you had a slide that you mentioned the increase of cutin being correlated directly to a better resistance in pests or infections.
Yeah. Well, there are not many reports on this topic, but what I read is that some of the organisms that colonize fruit can penetrate the cuticle because they secrete, they produce catinases. So I suppose that in these cases the resistance could be some sort of inhibitor of this cotinase activity. But actually I don't know. As far as I know, there is not much published research on this topic. But there is a very interesting work on Bolinha peach, which is a Brazilian cultivar, and it's very resistant to fungal attack, as far as I know. That could be a reason.
Thank you.
Welcome. Yeah.
So on the waxes and on the heating component of the cuticle of a plant, is that directly related to, or is it dependent on the sweetness of the fruit?
Is it related
Is it directly related on sweetness? Because some
to sweetness? No, I don't think so. No, not at all. All the contrary, because in some species you will find also variable amounts of phenolics, and phenolics, as you know, are not sweet, are astringent, which is also an issue for edible coatings, because if you apply an edible coating in order to, with the purpose of reinforcing cuticle, it may be very effective in this regard, but it could be that the taste of the fruit changes and is not good. Of course, many fruits you peel before eating, but many fruits you eat with peels. So we should have at some point, some sensory analysis also in order to check for this. Yes?
In one of your slides, you showed the different structure or the morphology of the cuticle in different varieties. Do you think that can be important or correlated to the stress tolerance?
Yes. When I was preparing this presentation, and I had all these results on different fruit types, I was again wondering about the very low amount of alkanes in olive cuticles. Oh, how can that be? It could be experimental error, but we found exactly the same in all 9 olive cultivars we analyzed. And those were the results. Of course, you cannot change them. But then when we analyzed the cutting, we found enormous percentages of hydroxy acids. And it has been reported that the hydroxy acids actually help the assembly of the cutting matrix. So it could be that the function of this hydroxy acid is enough to provide this resistance to water loss. Actually, in this study with the 9 olive cultivars, we found some cases in which the relative amount of hydroxy acids was around 80%. So I think that there is some probability that these hydroxy acids may be also important in this regard. Yes.
So, and another question from the audience. Regarding the post-harvest treatments that you've studied, are there any general positive or negative treatments, any do's or don'ts from a post-harvest treatment perspective?
Well, in particular quality attributes, you mean? Yeah. Yes, that that was one of the objective of our projects, and actually results were not what we expected. They were a bit poor, but yes, we could find some effect on water loss. So as I show in that summary, in that table, the ratio between alkanes and the cyclic compounds, the higher this ratio was in our studies, was associated to lower water loss. But this was an observation only on this attribute. For firmness, for instance, I don't remember that there were effects, or at least consistent effects. Sometimes yes, sometimes no. So, yeah.
Thank you.
Yes. Thank you.